Human cells essentially have a fingerprint, which allows T cells to recognize cells as being part of self and also to recognize cells which may have become cancerous or infected with virus. This is because all body cells with a nucleus and even platelets express the class 1 HLA genes, the human leukocyte antigens, which form this MHC complex, the major histocompatibility complex. These MHC proteins are actually a heterodimer involving proteins from the short arm of chromosome 6, one of the multiple HLA genes, and then a protein from chromosome 15, the beta-2 microglobulin. It is to this structure that a cytoplasmic peptide binds. The major genes, sometimes known as the classical genes, HLA-A, HLA-B, and HLA-C, are expressed on most body cells and are the most polymorphic genes known. They can possess thousands of different alleles. The great variety of these alleles guarantees that no new virus might be able to infect human cells and pass unnoticed. There are also minor HLA genes which can be expressed in specific cell types and are less variable. One, for example, presents to natural killer cells. Others are important for the tolerance of the mother's tissue in the placenta to that of the fetus. Since there are so many HLA alleles, each person who inherits one from each parent, on the chromosome 6 from each parent, thus has a unique combination which would allow the cytotoxic T cells to recognize their cell fingerprint as opposed to others. This third individual has inherited two of the same HLA alleles. That is possible because while many are extremely rare, some being observed only once, some are more common. Most individuals, however, have two different forms of each HLA gene, which they have inherited from their two different parents. This person's HLA genes allow the cytotoxic T lymphocyte to distinguish between the host cells which belong in the body and then the cells which do not, even if it is a transplant. If the HLA proteins are not a good match from the donor to the recipient, the cytotoxic T cells will recognize the transplanted tissue as foreign and destroy it.